Yeah, welcome everybody. Thank you, Rory, for the kind introduction. And thank you, Brainbox Initiative, for having me here today and for the opportunity to present my research on electric brain stimulation and brain connectivity during speech processing. Well, my fascination for the neural basis of speech and language processing goes back to my internship in clinical neuropsychology and my work with patients suffering from post-stroke aphasia during my PhD. As you may know, one of the most common causes of acquired language disorders in the brain, like aphasia, is stroke. And you see here on this slide one example. There is a beeping sound coming in. Is everything working, Rory?
Everything's okay, don't worry.
Okay, great. And now I have the problem again. Yeah, that's really sorry, I have again a problem here. Okay, where was I? Yes, sorry. Here, okay. What you see here in this image on the left side is an example of an ischemic stroke, so the occlusion of the middle cerebral artery. And what happens is that the blood oxygen supply here fails, and this causes brain damage in this area of supply of this artery. And you see that this causes this brain damage, and this leads to aphasia, a very devastating condition, because this affects multiple modalities of language use, namely not only speaking, but also understanding, reading, and writing. Historically, cognitive neuroscientists learned a lot from patients with aphasia about the functional specialization in the brain. The most famous examples have been reported by Wernicke and Broca because they could identify brain regions that are important for certain aspects of the speech processing, like production and comprehension. Later, Lichtheim found out that Broca's and Wernicke's area are connected by the arcuate fasciculus, and Geschwind detected other speech-relevant areas in the inferior parietal lobe here. Well, this was not the end of the story. The distinction between speech production and speech comprehension was not as clear as it was initially thought. Over the years, only a weak correspondence between brain lesion location and aphasia syndrome could be confirmed. In a most recent study shown here, published this month in Brain, it was shown that the effect of brain damage to Broca's area on speech production is to a large portion mediated by co-occurring damage to the anterior part of the arcuate fasciculus. This suggests that the connection within the network might be as important, if not even more important, than the integrity of individual nodes. As you see, the idea of the speech network has been incorporated by contemporary models of neural speech processing. Here, using the example of the dual stream model by Hickok and Purple, from this model you can take away that there are remote interconnected brain areas that are specialized for the processing of different or complementary aspects of the speech signal. One thing that I would like to emphasize is that successful speech perception in everyday listening is based on bilateral processing. Not only the left hemisphere, but also the right hemisphere is important, but with different specializations in each hemisphere. I just introduced the relevant brain network for neural speech processing, but how is information that is processed in remote brain areas eventually integrated? Or how does the brain bind information processed by different areas? Well, one idea is that through development, through learning and synaptic plasticity, the relevant brain areas are already structurally connected. However, structural connectivity might not be sufficient because cognitive functions require flexibility in routing of signals in the brain. A very popular idea is that the brain can build its own flexible communication structure on top of the anatomical connections by synchronizing oscillatory brain activity across the relevant brain areas. This idea goes back to the communication through coherence theory proposed by Basil Preisig. This theory suggests that only coherently oscillating neuronal groups can interact effectively because their communication windows for input and output are open at the same time. If we assume that the brain is using neural synchronization as a mechanism for interregional integration, what is the temporal scale that is relevant for speech processing in the brain? Before we address this question, let's see how we identify speech sounds. To understand speech, we need to integrate information that is conveyed over time. We can differentiate two important meaning-carrying features in the speech signal. One is the acoustic speech envelope that approximately corresponds to the syllable rate, and is here illustrated by this red trace that follows the sound pressure wave. The additional information is important additional information is provided by the temporal fine structure here illustrated in this speech spectrogram. The temporal fine structure of speech carries low-level features like phonemes, the smallest meaning differentiating speech sounds. The spectrogram you see in black has spectral energy concentrations here. And these spectral energy concentrations, they are caused by the acoustic resonance of the human vocal tract when we speak. And they provide very important cues to differentiate between different speech sounds. Interestingly, there is temporal correspondence between meaning-carrying features in speech and neural signals recorded from the brain when we listen to speech. And this is shown here on this right side image. The theta frequency corresponds approximately to the length of the syllables, and the gamma frequency band corresponds to the duration of phonemes. If we listen to speech, the brain starts to track the acoustic speech envelope, and this means that neural oscillations in the theta frequency range start to follow the amplitude of the acoustic speech signal. According to current models of neural speech processing, this represents a brain mechanism that helps to segment the speech signal into individual syllables. In parallel, it's thought that nested gamma band oscillations support the decoding of the temporal fine structure within the syllable chunks. There's always a noise coming in. I was not sure whether that is everything is working, but I think, Rory, it's still everything working, right? That's probably just the questions from the Q&A.
Yeah, everything's fine. Don't worry. I'm watching. Everything's okay.
Yeah, great. Well, in cognitive neuroscience, we often study the link between behavior and brain activity. You can say we study often correlations between task performance and neural patterns observed in EEG or fMRI. Of course, we do so because our methods have to be non-invasive or minimally invasive to be applied in healthy human beings. Nevertheless, we often would like to know if modulating or changing neural activation also causes a behavioral effect, because we would like to establish a causal relationship between brain activity and behavior. And that's why we use also non-invasive brain stimulation, because it's safe, it's not invasive, and it can be applied in healthy human participants. In my research, I want to change the oscillatory synchrony in order to see if this leads to relevant changes in speech processing. The brain stimulation technique that we use for this purpose is called transcranial alternating current stimulation, short tACS. tACS is a type of electric brain stimulation where a weak electric current is applied from outside the participant's skull. The principle behind this technique is to entrain the firing rates of neurons to the frequency of the alternating current. This is illustrated here, that you have this external current, and you see that the spikes of the neurons get entrained to this current. And indeed, it has been shown that this works not only in vitro, but also in vivo, and that tACS entrain the firing rates. However, tACS does not enhance the spike rate, so it can only modulate it to an external frequency. On the right side image here, you see the paradigm that we use to manipulate oscillatory synchrony. We apply tACS with different phase lags. This is shown here by this middle panel, and we apply it to different brain areas with these different lags. On top, you see the electrode configuration that we use. We use this concentric ring electrode montage. And in the middle, you have these experimental conditions where we have, for example, perfectly synchronized condition with a phase lag of 0 degrees, or we have this condition that we call the antiphase tACS condition, where we induce a phase lag of 180 degrees. On the bottom of the image, you see that you see the simulation of the electric fields on the brain, and you see that you see the benefit of this montage, that the electric field is restricted to a certain brain area. Of course, it's not so focused as focused ultrasound stimulation that we learned about last week. Now we can ask what would be interesting targets for the brain for such brain stimulation. Well, in my research, I focused on two routes of communication. One is the intra-hemispheric route, and one is the dorsal processing route here in the left hemisphere. So the intra-hemispheric route connects left and right auditory cortices through the corpus callosum. This connection is important for speech perception because acoustic processing is initially dominant in the auditory cortex contralateral to the ear of presentation. This means that acoustic information from our left and right ear is initially reaches different parts of our brain. Therefore, the inter-hemispheric connection is important for the brain to integrate auditory information from different areas. The second route, the left intra-hemispheric connection from auditory speech areas to motor speech areas, is important because it allows us to verbally repeat what somebody just said. You can imagine that this is very important during language acquisition and speech production, and maybe even for speech perception under challenging listening situations. Let's start with interhemispheric integration. On the left, you see a paradigm that is used quite frequently to study auditory integration across hemispheres. It's called the dichotic listening test. In dichotic listening, two syllables are presented at the same time to the left and the right ear. If participants are asked what syllable they perceive best, they report more frequently the syllable presented to the right ear. There is clear evidence that this is because the syllable presented to the left ear requires interhemispheric integration here, because it's first represented in the right auditory cortex and has then to be integrated with the language-dominant left hemisphere. Interestingly, there is evidence that the brain mechanism behind this integration process could be neural phase synchronization, and this is shown here on the right side. If participants report the syllable presented to their left ear, we see a significant increase here. Left ear reports are illustrated in blue. You see a significant increase here in neural gamma band phase synchronization. Well, a critical question is, of course, whether the observed increase in phase synchronization contributes functionally to the number of left ear reports. Or, in other words, what would happen if we could change neural synchronization? In our studies, we also used dichotic listening task, but with the difference that we did not present two syllables, but we presented an ambiguous syllable, a speech sound intermediate to the syllables ta and ga, to the right ear, while a disambiguating cue was presented to the left ear, with the idea that participants integrate the inputs presented to the right and the left ear. This disambiguating cue contains an acoustic feature that differentiates between the syllables da and ga. The combined presentation of the ambiguous syllable and the disambiguating feature elicits a unified percept. And this you can see on this right side image here, namely that depending on the presented feature, either we presented this feature or that feature, participants perceive da or ga. This means that participants integrate the information of the feature. Again, acoustic processing is initially dominant in the auditory cortex contralateral to the ear of presentation. This means that the acoustic feature that is initially processed by the right hemisphere here has to be integrated with the ambiguous speech sound that is processed initially by the left hemisphere. To test the relationship between neural synchronization and binaural integration, we experimentally manipulated oscillatory synchrony with tACS. For this, we used high-density gamma band tACS with the conditions that I already introduced, the in-phase tACS or the anti-phase tACS with an inter-hemispheric phase lag of 180 degrees. In addition, we concurrently assessed effective connectivity between bilateral auditory cortices using fMRI and dynamic causal modeling, short DCM. In DCM, the strength and the direction of regional interactions are computed by comparing the observed fMRI time series in a given region, this one here, with the fMRI time series that can be predicted by a neurobiologically plausible model. This neural model here describes how activity in and interactions among regional populations are modulated by our tACS conditions. In our analyses, we focused on the effect of tACS on interhemispheric connectivity. Before I show you the connectivity results, I would like to show you the univariate fMRI data. On the left, you see that, that we observe significant bilateral activation in the auditory speech areas during syllable perception. These areas include also the primary auditory areas like Heschel's gyrus. And you see that these areas get activated across all experimental conditions here. If we use anti-phase tACS, in-phase tACS, or sham, we see this activation. On the right, you also see that tACS modulates local brain activity here in the right posterior superior temporal sulcus. This area is beneath the center of the annular tACS electrode. And this area is also involved in task performance because participants who activate this area more strongly were also integrating the acoustic feature more frequently. So there was a positive correlation between the two. Now let's look at the connectivity results. First, I would like to show the effective connectivity data without brain stimulation, so the connectivity data during sham. On the left, you see the areas that were included in the DCM analysis here. You see bilateral Heschel's gyrus in yellow and bilateral posterior superior temporal sulcus in black. You also see our neural model, and the numbers on these arrows represent the coupling parameters in Hertz. Positive numbers in green mean that area A has a positive influence on area B, so that area A is causing an activation increase in area B. If you have a negative connection, this area here induces an activation decrease in the other area, so just the opposite. What we find is that we have positive bidirectional interhemispheric connectivity for Heschel's gyrus, and this reflects just mutual bilateral activation increases and decreases over time. On the right, you see that we observed a positive correlation between interhemispheric connectivity and binaural integration. So participants with higher bidirectional connectivity also show higher binaural integration scores. This suggests that inter-hemispheric connectivity contributes to binaural integration. What are the effects of tACS? We find that tACS modulates both intra- and interhemispheric effective brain connectivity. Here you see in red the effects of in-phase tACS and in blue the effect of antiphasic tACS. So both tACS conditions had negative modulatory influences here on intrahemispheric connections, but only antiphase tACS significantly reduced interhemispheric connectivity between bilateral Hessel's gyrus. Sorry. On the right, you see that we found a correlation between the strength of the modulation induced by antiphasic tACS and the strength of the tACS modulation on the connection strength and the modulation of tACS on binaural integration. So the stronger the induced interhemispheric connectivity reduction, the stronger the reduction in binaural integration. I would like to emphasize the phase selectivity of the gamma band tACS effect that we observed here. Antiphase but not in-phase tACS reduced interhemispheric connection strengths between the auditory cortices. We propose that the interhemispheric phase difference induced by the antiphase condition induces a lag in the relative phase of the underlying cortical areas, and that this lag is responsible for the reported change in interhemispheric connectivity. The observed changes in binaural integration suggest that the induced lag is maladaptive for information transfer, consistent with the idea that neuron groups that do not undergo coherent excitability fluctuations communicate less effectively. However, not only antiphase tACS perturbs binaural integration, but also in-phase tACS. And this is because in-phase tACS reduces intrahemispheric connectivity. Something else that is important to consider, not all participants show the same susceptibility for tACS. In our study, participants with higher binaural integration scores were more likely to show a reduction in binaural integration in response to tACS, whereas participants with lower binaural integration were more likely to show an increase in binaural integration. A similar result was reported in 2016 by Ruffiner and colleagues. They showed that gamma tACS perturbed speech perception in young participants with good performance. However, in elderly participants with lower performance, gamma tACS enhanced speech perception. This means that the behavioral effect of tACS may be positive or negative depending on whether a participant has a good or bad baseline performance. So this might be because participants with good performance who show an optimal level of gamma band oscillatory activity, that there the tACS induces additional noise that makes performance worse. However, in participants with lower baseline performance, tACS might either reduce noise by inducing synchrony and thereby boost performance, or increase noise, thereby making the stimulus-induced brain signal detectable through stochastic resonance. So what is important, what you can take away from this, that there might be an inverted U-shaped relationship between the for the effect of tACS on behavior. What are the implications of this study? Well, I would say our results support the idea that neuronal phase synchronization between different brain areas is fundamental for neural integration. Our results give us valuable insights, for example, how we can use brain stimulation techniques to manipulate connectivity between different brain areas. This could be a first step to test this in other sensory modalities, for example, or with more complex auditory stimuli. The results might also be interesting because of their clinical implications, because altered inter-hemispheric connectivity has also been reported in tinnitus and auditory verbal hallucinations. However, the causal relationship between these syndromes and brain connectivity is still unknown, and if we are able to manipulate connectivity, this could of course help to find out more about this relationship. Yeah, and this study was recently published in PNAS, and if you are interested to read it, that's the link to this study. Well, in the last part of my talk, I would like to present you a study now where we aimed to translate this also to speech production. So we wanted to modulate neural phase synchronization during speech production. In the introduction, I mentioned that sensory and motor speech areas are interconnected by the dorsal processing route here, and that includes the arcuate fasciculus. Auditory-motor mapping is the process by which sensory acoustic information processed here in the auditory areas is mapped onto articulatory-motor representation that are relevant for speech production. Auditory-motor mapping is the process that allows us to verbally repeat what somebody just said, and that is presumably very important for language acquisition and maybe also for speech perception under challenging listening conditions. Recently, it has been found that the neural phase synchronization between sensory and motor speech areas during speech perception is dominant in the low theta band. This is very interesting because this rate is consistent with the average syllable rate across languages. In this study, we wanted to find out what would happen if we modulated oscillatory synchrony between sensory speech areas and motor speech areas in the left hemisphere, while participants perform a verbal repetition task. We applied tACS, this time over sensory and motor speech areas in the left hemisphere, while participants were listening and repeating trisyllabic non-word stimuli. I will try to play the sounds for you here. I don't know, Rory, you can tell me if it works. Did you hear a sound?
I didn't, sorry.
Okay, then it's not working. So that were rhythmic. Then I described the stimuli. It's word rhythmic, trisyllabic non-word stimuli composed of consonant vowel syllables, legal syllables from the Dutch language. In black, you see here the sound pressure wave of the syllable, and in gray, the frequency matched noise that we added to make this task harder for the participants. tACS stimulation was applied this time at 4 Hertz, and this was the same rhythm as the syllable presentation rate of the non-word. Again, we had these 2 main stimulation conditions, in-phase and anti-phase tACS. And you see the sinusoidal currents, and the color gradient here represents different phase alignments between the tACS current and the non-word stimulus, because for this low frequency tACS, it has been shown that there might be different phase alignments where participants have best performance or are most susceptible to the tACS stimulation. So what happened? Unfortunately, not much. We did not see a stimulation effect. And you may see that in this graph here. We found no difference across the stimulation conditions in phase, anti-phase, and sham stimulation. And you see here this gray area, this gray shaded area, that's just the average performance across conditions. We also tested whether there's an effect of the relative tACS non-word phase. On the x-axis here, you see the task performance as a function of the distance from the participant's best lag. So the best lag represents the lag where participants show best performance. That is usually considered because this is varying across participants. So some participants that show a best lag at a different time. So the data of individual participants in these studies is then usually phase wrapped to the peak and to this peak here. And if you do phase wrapping, the peak becomes trivial, of course, and therefore has to be excluded from the analysis. Here you see a behavioral time series that we would expect if we see an effect. And you can still, if you exclude the peak, analyze then whether the time points that are close to the peak show better performance than the time points that are close to the trough of the behavioral time series. But that was clearly not the case here. So we did not find a modulation of the interareal phase difference, but also not of the phase alignment between the tACS current and the stimulus presentation. Well, with this, I'm already at the take-home message. Well, can we use electric brain stimulation to alter connectivity in the speech network? I would say tACS has potential, but it's still underexplored in speech research. And therefore, it is also important to know what is working, and it's very important to report negative results. We have observed some limiting factors. Perception seems to be more susceptible than speech production. One reason might simply be that we are limited in terms of stimulation intensity with tACS, so the applied currents have to be weak, because otherwise participants would feel the stimulation and blinding would not be possible anymore. I think that technical advances will hopefully allow us to use more multimodal approaches in future. I think that concurrent neurophysiological recordings like the fMRI recording we used are very important, because they help us to interpret unexpected stimulation effects. Like in our example, we did not expect that in a phase tACS would change intra-hemispheric connectivity. If you want to start electric brain stimulation, what can you expect? I would say that we can expect small effects, but these are nevertheless interesting effects because they help us to understand the mechanistic roles for tACS, of course, of the oscillatory brain activity. With other techniques, we can study other mechanisms in the brain. But we also need to have a good understanding of the brain feature that we would like to target, and we need very clear and concise hypotheses. Before I end my talk, I would like to show you some ongoing research on sensory-motor integration that we are currently conducting in our lab. Previous research indicates that sensory-motor integration may also play a role in speech perception, as I mentioned beforehand. For example, if the speech signal becomes less comprehensible because we degrade it, we reduce its frequency resolution, we can see additional activation of the motor cortex when participants try to understand these speech signals. Moreover, the perturbation of the articulatory-motor cortex with transcranial magnetic stimulation reduces not only the excitability of the articulatory-motor cortex, measured here with motor-evoked potentials, it also perturbs phoneme identification and discrimination. In our lab, we are currently investigating if we can achieve the opposite, namely whether we can enhance the excitability of the speech motor cortex with kilohertz tACS, and whether this will boost speech perception. If we achieve to modulate, for example, degraded speech perception in normal hearing listeners, then this might be also beneficial for patients with hearing impairments. With this, I would like to end my talk here. The work that I presented today was conducted during my fellowship that was founded by the Swiss National Science Foundation, and the fellowship I did at the Dons Institute and at the Max Planck Institute for Psycholinguistics in Nijmegen in the Netherlands. I would like to thank all the people who made this work possible. I would like to thank my lab, headed by Alexi. I would like to thank my international collaborators and the research assistants, of course, and the technical group. Special thanks to Benjamin Kopp. to Birgit Knudsen and Iris Schmidt, who tested participants that for these studies that I presented today. And of course, I would like to thank you for your attention. And now I'm happy to take your questions. Thank you very much.
Thanks, Basil. That was fantastic. We'll just give some time for the questions to come in. And in the meantime, I'll ask you one. You mentioned that like inverted U-shaped curve and the relationship that may have with ongoing gamma oscillations. Is that correct?
Yes, exactly.
So I'm just making sure I understood you. Do you think it might be useful to do things like EEG to obtain individual gamma oscillations and then apply that frequency of tACS to them rather than applying a fixed frequency to all participants?
Yes, that is of course a very good approach, and it has already been taken in other frequency bands. I think here of the alpha oscillations. There people are usually measuring the resonance frequency of the system first with EEG, and then they apply it to the individual alpha frequency. That is also something that I plan to do in my future research. Another thing, of course, is with EEG, it could also be interesting to measure first the subjective inter-hemispheric lag that participants have, and then target this particular lag to see how this influences integration.
Cool. Thanks for asking the question. So we don't seem to be, no questions are coming up as of yet. So we'll just give it a few more moments, and if not, we could round it up here. You also mentioned the idea of dorsal and ventral streams, and a while ago a paper was published showing that dorsal was all about action and ventral was all about perception. Do you think that dichotomy still stands now?
Well, it is an interesting model, and I think it's it was not it was first formulated for visual perception and then translated to speech perception. And in speech perception, that probably did not emphasize is that it is really for it is thought for novel stimuli that just need to be imitated. So for example, non-words. And if you have meaningful stimuli, it is thought that the ventral route can also be used to repeat them. So that means that patients with aphasia, for example, even if their dorsal stream is lesioned, they may not be able to exactly repeat what they heard, but to use synonyms or to use another just to explain what they heard. Yeah.
Great. Thanks for answering that. So I've got a couple of questions coming in now. One of them is saying you were testing auditory integration. Would there be any way of using tACS for audio-visual integration?
Yes, I think so. I'm not aware of a study right now that did that, but there are interesting things with looking at the phase of neural oscillations and how this influences auditory perception. And it has been shown that for example, the auditory cortex to some extent is enslaved by the visual cortex, so that the phase of visual oscillations have a big influence on the auditory cortex, and that, for example, that lip movements cause phase resets in the auditory cortex and this further affects the speech perception.
Nice. Thank you for answering that. And then we've got one more question that's come in. Do you think there is a way to qualitatively assess the conduction speed of the pathway interconnecting each hemisphere's auditory area?
Yes, I think so. That has been done. I think it's a simple EEG experiment that is usually done, that you present a tone, and then you measure the latency of the ERP components on the left and the right hemisphere, and it has also been shown that there is considerable variance across participants, and there seems to be differences in clinical conditions like schizophrenia, where this intrahemispheric transfer is altered.
Nice. Sounds like a cool sensitive measure. So we don't have any more questions coming in now. I'll move on to the summing up part now, where I talk about what's coming up next. But first of all, I'd just like to thank you, Basil, for that talk. It was really interesting, really cool work using. I do like the concentric ring with the disc electrode approach for the high definition. I think it's definitely the way forward for more precise applications of tACS, and even doing stuff in and out of phase tACS. That definitely looks like it's the